So today we're going to do the second part of protein synthesis called translation. The next few slides will be just review. There is a couple of things that you should write down, but not too many. So remember, DNA stands for deoxyribonucleic acid. We've gone over that multiple times. Please remember what it means. And both DNA and RNA are nucleic acids. They have nucleic acid in their name. And they are long polymers of nucleotides. Nucleotides are the monomers of nucleic acids. So remember that the outside of DNA is called the sugar phosphate backbone, and this gives the DNA its shape and structure. Here you have our phosphate and sugar. And the nitrogen bases are on the inside of the double helix, and it's the sequence of these nitrogen bases that gives us the information. So these give us our information for our body and proteins that need to be made. Remember that a gene is a section of DNA used to make proteins, and these genes are sections of DNA on the chromosome that carry the useful information. All of our chromosomes are called the genome, which is a complete set of DNA containing all the genes in the cell. Let's review a little bit about transcription. So transcription, remember, makes copies of the DNA. And in order to do this, the DNA unwinds and separates between the nitrogen bases to expose the nitrogen bases which carry the information. Once the strands of DNA are separated, the enzyme RNA polymerase copies information from DNA to a newly made strand of RNA. So, there are 3 different types of RNA that are involved in making the protein. The first type we've discussed, which is mRNA, or messenger RNA. This is the RNA that brings the message from DNA in order to make the protein. Next, we have transfer RNA, and these bring the amino acids in order to build the protein at the ribosome. And the last one is ribosomal RNA, which is the ribosome or rRNA. And that's the one that actually puts the protein together. So in eukaryotes, many proteins are made by the ribosome in the cell protein factory or the rough endoplasmic reticulum. So if you see here, the rough endoplasmic reticulum has a bunch of these little red dots, and those are ribosomes. So we call the rough ER the protein factory. We also have free ribosomes that are just around the cell in the cytoplasm. So remember, the mRNA is made during transcription. After transcription happens, that mRNA leaves the nucleus and attaches to a ribosome. The protein is built according to the universal genetic code. So the universal genetic code is common to all, from the bug on the floor to the horse in the stall. And just remember that all living things will use that same genetic code. So basically saying that all living things will have A, T, C, and G in their DNA. So an important word to remember is codon, and it's a set of 3 nitrogen bases. Each codon is translated into an amino acid in the protein. So the codons are on the mRNA, and those get translated, and an amino acid will be brought for every 3 of those nitrogen bases. So transfer RNA or tRNA can base pair to the codon, and it enters a ribosome and attaches its amino acid to the growing protein. So ribosomes are organelles that direct the construction of proteins. They read the mRNA and use the amino acids provided by tRNA to build the proteins 1 amino acid at a time. These proteins that are built might need to be modified, so they go to the Golgi apparatus, the Golgi body, where sometimes lipids and sugars are often attached. So let's discuss gene expression. So using the information in genes of DNA to make products in the cell, that's what it is, and it's a regulated process. So basically it's saying that the cell turns genes on and off so that only certain genes in the cell are expressed. So this regulates how genes are expressed during transcription. Basically meaning you have a lot of information, but not all of that information is going to be used. Not everything will result in a protein. So if they are off, they are unexpressed and no product is made from that gene. Cells can also regulate how a gene is expressed by altering the mRNA before a protein is made. So cells can splice the mRNA into different combinations that produce different proteins. And here we see that process happening. So this controls how genes are expressed during the process of translation. However, this is a very specific process of regulating gene expression that only happens in eukaryotic cells. So bacteria will not be doing this. So using a codon chart. In order to decode the mRNA sequence, we need to use this universal codon chart. We have one form here, another form here, and almost every amino acid has more than one codon. So you can make serine in different ways because we have UCU, UCC, UCA, UCG, and this prevents mutations from changing proteins. So what is one of the correct codons for each of the following amino acids? For proline, we might have CCU, CCC, CCA, or CCG. For serine, we might have UCU, UCA, AGU, UCC, UCG, AGC. Alanine, we might have GCU, GCA, GCC, or GCG. Methionine, one of the rare ones, only has AUG. Cytosine can have UGU or UGC, and arginine has CGU, CGA, AGA, CGC, CGG, and AGG. So now if we were going to translate this mRNA, and we know it's mRNA because it has U's in it, into the correct amino acid sequence, we would start off by reading 3 letters at a time. So CUG is our first codon, and then ACC, and then UAC, and then AUA. So putting a line after every third letter helps you make sure that you're reading the correct codon. So if we were to look back at our codon chart, CUG would code for leucine, ACC would code for threonine, UAC would code for tyrosine, and AUA would code for isoleucine. So now we're going to code again. Again, we put our lines after every third letter, and then we code. So AUG will give us methionine, GGU will give us glycine, and then UGA gives us a stop. So that stop means that we stop making our protein. The codons after that do not matter. So stop means stop. And these stop codons, so that means no tRNA is attached and the production of the protein stops. The finished protein is released from the ribosome. And here we have the stops in the codon chart. Okay, those are the notes for today. Make sure that you wrote everything that was in red and green, and I'll see you tomorrow.